Hello everyone, myself Dr. Girisha, working as assistant professor in Department of Plant Pathology, School of Agriculture, SR University. Today I'll discuss about rice disease and their integrated management. So this is the content of this PPT. First we'll discuss about fungal disease, then bacterial disease, then viral disease, then nematode disease and physiological disorders of rice. Let's first see about the brief introduction about the rice crop. The rice is a member of Poaceae family. Rhiza sativa is believed to be associated with a wet, humid climate, even though it's not a tropical plant. Rice is an important staple food crop, more than 60% of world population. India stands second position after China with respect to rice production. The world estimated area with respect to rice production is 496 million metric ton with an area of 158 million hectare during 2016. These are the list of fungal disease, bacterial and viral disease in rice. First, we'll see the list of fungal disease: blast of rice, brown leaf spot, sheath blight caused by Rhizoctonia solani, stem rot caused by Sclerotium rhiza, foot rot of rice caused by Fusarium moniliforme, false smut of paddy caused by Ustula genodia virans, then Udbata disease of rice caused by Apellis oryza. Then coming to list of bacterial disease, bacterial blight of paddy caused by Xanthomonas oryza, Pithover oryza. Then bacterial leaf streak of paddy caused by Xanthomonas oryza, Pithover oryza cola. So these are the list of viral disease. One is the rice tumor virus and grass stunt of rice. Coming to nematode disease, rice white tip nematode, which is caused by Aplycoides bessi, rice root knot nematode, Meldogena graminicola, Ufra disease of rice, Didelyncus angustus, and physiological disorders in rice like Kyra disease of rice, which is caused by zinc deficiency. First, let's start with the blast disease of rice. This blast is caused by the fungus Pyracularia oryza. It will able to cause more than 70 to 80% damage under severe condition. And in India, this is first time reported in 1918 in Tanjavur. It is affecting more than 80 countries in the world. And disease is more severe in high rainfall area and the areas which are receiving more nitrogen fertilizers. Coming to the symptoms of rice blast on the leaves, the lesion appear as a small bluish green flecks. The lesion soon enlarge under moisture weather, form a characteristic spindle-shaped spot with a grayish center and dark brown margin. So this is the peculiar symptom of leaf blast. Like the same spots occurs in the nodal region, then girdling of the nodes occur that will result in the nodal blast. So I already see these are the symptom of leaf blast. Disease can infect like all the growth stages, including the leaf region, neck region, and node region. Among the three leaves and neck blast, it will cause severe infection. Like already explained, small specks originate on the leaves, subsequently enlarge into spindle-shaped spots with a zero 0.5 to 1.5 centimeter in length and 0.3 to 0.5 centimeter in width with a ashy center. Severe spots they will coalesce that will result in big irregular patches that will look like leaf blast appearance. Severe case of infection, the entire crop gives a blasted appearance or burnt appearance, hence the name blast. Then coming to other symptom like a neck blast, the neck region of the panicle develops a black color and shrivels completely or partially. The grinds that may be inhibited, a panicle break at the neck and it will hang down from the main stem. So this neck blast, it will cause severe losses in case of paddy blast because once the neck or panicle is infected, there is no development of panicle or there is a development of choppy grains or unfilled grains due to lack of supply of photosynthetes like sugars, carbohydrates from leaves region to the developing neck region. Coming to nodal blast, already explained, the nodes become black and they will break up. Sometimes there is a lodging also occurs. So this is the fungus etiology. This pathogen belongs to phylum Ascomycota and the sexual stage of the fungus is Magnoporta oryza. This sexual stage of the fungus will produce perithecium. These are the conidia. We can call it as a pyriform conidia with a three-celled pyriform shaped conidia. Coming to the life cycle or infection cycle, this blast disease is mainly seed borne in nature. When the farmers sow infected seeds for sowing, there is infection to the young seedling. Then young seedling, it will develop the disease. Then it will spread the disease to other healthy plant, and there is a transmission of the disease, the pathogen mainly through infected conidia, like wind-borne conidia or hair-borne conidia. Coming to epidemiology of rice blast, the plants of the rice are very susceptible at the seedling stage and rapid tillering stage and ear emergent stages. So these are the three stages of the rice which are susceptible to blast attack. Condition conducive for rice blast include the long periods of free moisture or the leaf wetness is required for the infection, excessive application of the nitrogen fertilizer that will favors the disease. So when we apply excessive nitrogen fertilizer, it will reduce the thickness of the cell wall. Pathogen can easily penetrate. Minimum night temperature ranges from 20 to 26 degree along with higher relative humidity of 90% for a 1 week at a susceptible stage of the crop. There is an outbreak of the blast disease. Condition that favors the development or sowing of the disease seeds, poor airflow, poor sunlight penetration, and rainy days with a high humidity. All these conditions are highly favorable for the blast incidence. Next, coming to management of rice blast, mainly through we need to avoid application of excessive nitrogen fertilizer. Apply the nitrogen in 3 split doses. Remove weed wash from the bund region. Clean cultivation. Use of tolerant varieties like Penna, Pinakini, Tikana, then IR series, Prabhat, Swati, Jaya rice, which are resistant to tolerant to paddy blast. Burning of straw and stubble after harvest. Dry seed treatment with pseudomonas fluorescens. Dark formulation we can apply at the rate of 10 gram per kg of seeds. Seed treatment with same like fungicides like Captan or carbendazim or Thyram or tricyclozole at the rate of 2 gram per kg. We can also go for spraying of pseudomonas fluorescens stock formulation at the rate of 0.5% from 45 days after transplanting at 10 days interval. Spraying of tricyclozole at 1 gram per liter of water or edifenophos at 1 ml per liter of water, or we can also apply carbendazim at 1 gram per liter. Like tricyclozole is highly effective fungicide against the rice blast. Then coming to another important disease in rice, which is brown leaf spot of rice. This brown leaf spot of rice, which is caused by Elementosporium oryza. The disease is very common in rice growing area. Epiphytic form of disease occurred in West Bengal in 1942. You know the famous Bengal famine, which is caused due to brown leaf spot of rice. The disease in soil and seed borne in nature, and disease seeds show poor germination capability. Extent of yield loss may range from 50 to 90% in case of extreme infections. This is the Bengal famine during 1942 and 43. You know, millions of people died in India due to single disease, brown leaf spot of rice. Coming to the brown spot of rice, you know, this occurrence also called as sesame leaf spot, Elementosporium or fungal blight, mostly seen in West Bengal, Orissa, AP and Tamil Nadu. Almost all parts of India, the disease will affect the rice crop. Coming to the symptom, occurrence in nursery as well as main field, causes blighting of the seedling. Leaf spotting is very common symptom. Isolated brown round to oval, resemble like a sesame seeds. Spots measure about 0.5 to 2 mm in breadth, collision to form a larger patches on the leaves. Coming to other symptoms, the seeds will also get infected. Black or brown spots on the glume spots are covered by a olivaceous velvety growth of the fungus. Infections also occurs on panicle neck with a brown color appearance. 50% yield reduction in severe cases may occur. So in the infected images, we can see the sesame spot or sesame seed like brown color spots with a dark brown margin. And the same kind of spots we may also seen in the affected seeds also. Because coming to etiology of the fungus, this will belong to the phylum Ascomycota, the genus Elementosporium, and the sexual stage of the fungus is Cocculobellus myabenis. It will produce very 3 to 4 slightly curved conidia. Coming to disease cycle, no, this disease is both seed borne and soil borne. Mainly the disease spreads through seed borne in nature. Coming to epidemiology of brown leaf spot of rice, the disease occurrence was favored in a dry environment whereas reduced amount of water was present. Humidity level of the leaves and lowered minimum temperature for the crop cultivation favors its epidemics. The optimum temperature including 28 degrees Celsius is highly required for the brown leaf spot. If the soil minerals such as nitrogen, potassium, phosphorus, silicon and manganese are deficient, then we can see there is a occurrence of brown leaf spot. Brown spot of rice is favored in areas where water level is low in soil. So these are all the epidemiological conditions for the brown leaf spot of rice. Coming to management of brown leaf spot of rice, one is it's a seed-borne disease. Go for disease-free seeds for sowing. Since seeds, we can soak the seeds or seed treatment with Captan or Thyram at the rate of 2 gram per kg of seeds. Then also application of carbendazim or mancozeb or edifenophos at the rate of 2 gram per liter at 10 to 15 days interval. Spray preferably during early hours or afternoon at the flowering and the post-harvest stages of the crop. We can prevent the infection. So that's regarding brown spot of rice caused by Helminthosporium oryza. Then coming to sheath blight of rice. So the sheath blight of rice, it is caused by Rhizoctonia solani. Rice sheath blight is an increasing concern in rice production. Nowadays, the disease is severe in occurrence. Yield loss of 25% was reported at the flag leaves are infected. In the United States, yield loss up to 50% is reported. Sheath blight is also caused yield loss up to 6% in tropical Asia. Coming to the symptom of sheath blight of rice caused by Rhizoctonia solani. In the initial lesions are water soaked to greenish grey and later become greyish white with a brown wavy margin. Legends of the leaf sheath near the water line, presence of sclerotia in the soil, the floating sclerotia coming in contact with the leaf sheath, then the leaf sheath will infected, then we can see irregular greenish grey wavy brown margin. Then the lesions will coalesce, that will gives like a blighted appearance. And also another symptom is partially filled grains or empty grain is the character of sheath blight of rice. So this is the images representing the different symptoms of sheath blight. The first picture we can see sheath blight symptom like a typical sheath blight symptom and formation of sclerotia on the leaf sheath. Coming to the sclerotia, which is a vegetative structure of the fungus, which is mainly formed by aggregation of hyphae or aggregation of mycelium. Then it will converted into a dark black like structure. It is like a main survival structure of Rhizoctonia solani. Coming to pathogen character, this Rhizoctonia solani and this Telomorphes Thanadophorus cucumaris. The Rhizoctonia solani frequently exists as a thread-like growth and plant. In culture, it considered as a soil-borne pathogen. The pathogen is not currently known to produce any asexual spores or conidia, but it mainly survives through, as I explained, macrosclerotia. It produces white to deep brown mycelium when grown on the artificial medium. The fungus produces macrosclerotia, and rectangular branching of the mycelium is the one of the most identifying character of this fungus. Coming to taxonomy, this phylum belongs to Basidiomycota, like class Agaricomycetes, order Canthariales, family Ceratobasidiacea. Coming to the life cycle of the sheath blight or disease cycle of sheath blight of rice, firstly we can see the floating sclerotia present in the soil. They will, when they coming in contact with the sheath like a leaf sheath of rice, then they will get germinated and they will cause different symptom like wavy brown margin, greenish to grey color, wavy brown margin, blast or blighted appearance on the leaf sheath. Then this floating sclerotia, again they will released into the water, again they will carry it through one field to another field through irrigation water. So mainly the fungus is survived by production of dormant macrosclerotia. So this is regarding the disease cycle of sheath blight of rice. Coming to favorable condition, sheath blight occurs with a high temperature of 28 to 32 degree with a high level of nitrogen fertilizer and relative humidity of 85 to 100%. Plants are more vulnerable to sheath blight during rainy season. High seedling rate or close planting, dense canopy, disease in the soil, sclerotia, infectious bodies floating on the water, which will have the favorable condition for the sheath blight of rice. Coming to management, application of FIM 12.5 ton per hectare or green manure 6.25 ton per hectare promote the antagonistic microflora. Soil application of pseudomonofluorescence at the rate of 2.5 kg per hectare mixed with the 50 kg of FIM after 30 days after transplanting. Then foliar application of same pseudomonofluorescence at the rate of 0.2% at the boot leaf stage. It can prevent the disease. Avoid the flow of irrigation water from infected to healthy leaves. So all these are different management strategies to avoid the sheath blight infection. Coming to chemical management, like application of carbendazim 1 gram per liter or propiconazole 1 ml per liter, we can apply to control the fungus. Spraying of the infected plants with the fungicide such as binomil, iprodin, antibiotics such as validomycin, polixin is effective against the disease. We can also reduce the nitrogen application, or we can skip the top dressing with the nitrogen. So all these are different management or IDM strategies to control the sheath blight of rice. Then coming to false smut of rice, one of the most important fungal disease. The false smut of rice or palfasma paddy, which is caused by Eustillaginodia virens. The disease affects at the early flowering stage of the rice crop. The ovary is destroyed. The second stage of infection occurs when the spikelet nearly reaches maturity. These causes chalkiness and can reduce the 1000-gram weight in seeds. It causes reduction in the seed germination up to 35%. The disease can severe and it can lead to loss of 25%. In India, yield loss may go up to 7 to 75% under severe condition. Coming to the symptom of false smut of rice, ears individually ovaries are transported into large velvety green masses. These are round to oval, attain the size of 10 mm in diameter. Only few spikelets are infected. Glooms are not affected but covered by a membrane. Color changes when it is exposed by breaking of membrane. This is the life cycle or disease cycle of the false smut of rice. Majorly the fungus will produce chlamydospore. The chlamydospore play important role in the secondary infection, which is a major part of the disease cycle. Chlamydospore form on the spore ball, which is spherical, elliptical, warty, volvacious, with 3.3 to 5 and 4 to 6 micrometer in diameter. Younger spores are smaller, paler and also smooth. Some of the green spore ball develop on the 1 to 4 sclerotia at the center. These are the sclerotia overwinter in the field condition. These sclerotia are the spore ball. They contain nothing but a chlamydospore. In the temperate regions, the fungus survives in the winter by means of sclerotia as well as chlamydospore. It is believed that primary infections are initiated mainly by the ascospore produced from the sclerotia. This sclerotia, which is containing ascus and ascospore, which will cause primary infection. The secondary spreading or survival structure is the chlamydospore. These chlamydospore mainly survives in the soil. Coming to taxonomy, this fungus will belong to phylum Ascomycota, then class Sardidiomycetes, order Hypocoreales. Then coming to the genus Eustillaginodia virens. This is the disease cycle of false smut of rice. Here we can clearly see the smut infected paddy or paddy grains, which mainly contain chlamydospore. These chlamydospores produce on the surface of the smut ball, which will act as a secondary spreading agents, which can also survive in the soil for more duration and they will cause infection. Then the smutted spore containing ascospore and ascus, they will cause primary infection. Coming to management, use of disease-free seeds that are selected from the healthy crop, seed treatment with the carbendazim, control of insect pest, split application of nitrogen is recommended, removal and proper disposal of the infected plant debris, spraying of COC at the rate of 2.5 gram per liter, propiconazole at 1 ml per liter. At the boot leaf stage and milky stages, we can recommend to control the fungal infection. At tillering and free flowering stages, spray hexaconazole at the rate of 1 ml per liter or chlorothalonil 2 gram per liter is recommended. Coming to the next disease, Udbhatta disease. This Udbhatta disease of rice, which is caused by the Epilis oryza, or perfect stage is Balantia oryza. Also called as Agarbatti disease or Karikatti roga or Chipsuvade roga in Karnataka, in Kannada. This disease is endemic and minor importance in certain areas of India. The fungus causing Udbhatta disease was described from India in from India in its anamorphic stage as Apellis oryza. It is reported occur in severe form many parts of South India, including Madurai, Tamil Nadu, Wayanad, Kerala, Kollegala districts in Karnataka. Coming to the symptom of Udbata disease, a panicle emerges from the leaf sheath in a straight, dirty colored, hard cylindrical spike, much resemble an agarbatti or udbatti, hence the name agarbatti disease. The spikes infected by Apellis oryza are somewhat dry, mummified with a partially formed buds. No grains are formed on the panicle. Coming to the classification, the phylum belong to Ascomycota, class Aderiomycetes, order Hypocreales, family Claviseptaceae. Coming to the genus Apellis oryza. Coming to the disease cycle, the fungus is a seed-borne, externally seed-borne, and it will cause systemic infection. The presence of lustrous grayish white films of the fungal growth on the young leaves of infected seedling suggests the entry of pathogen during germination of the seeds. As no grains are obtained from the affected head, the disease seeds are not important for the disease occurrence. The fungus has been recorded on the grasses like Sanodon, Dactylan, Penicitum, other wheat species present on the rice cultivation act as a collateral host. The disease incidence is severe on very early and late some crops. These are the pictures representing the culture of Balantia Apellis oryza or Balantia oryza. These are the conidae of Apellis oryza. Coming to some factors which favor the disease like warm temperature, high humidity, early stage of planting from maximum tiller into panicle initiation stages are highly congenial for infection. Coming to management, use of disease-free seeds for sowing, seed treatment with a Captan or Thyram at the rate of 2 to 3 gram per kg, hot water treatment for the seeds at the rate of at the temperature of 50 to 54 degrees Celsius for a duration of 10 minutes. Solar treatment of seeds is effective in killing the pathogen carried in the seeds, if any. It is advisable to avoid the use sowing the seeds from the infected field. Removal destruction of the disease panicles is highly recommended. Coming to another important disease in rice, which is Buchanay disease of rice. This Buchanay disease, which is caused by Fusarium moniliforme, perfect stage is Gibberella fusicora. Earliest the known report of Buchanay disease from 1828. It was first described scientifically in 1898 by Japanese research scientist Hori. Crop losses may range from 20 to 50%. Coming to different symptoms of Buchanay disease in rice, disease occurs in nursery and main field. The chlorotic pale, thinner and abnormally elongated nodes we can observe. Tall, lanky plants in the field. Adventitious roots from the nodes near the ground level. Pinkish brown sporulation of the fungus at the base of the plant. Coming to the pathogen, the fungus infect plant through root or crones. It later becomes systemic in infection. It grows within the plant but does not systemically infect the panicle. The microconidia and mycelium of the pathogen are found to be concentrated in the vascular bundle, particularly at the large pitted vessel and the xylem tissue. The pathogen produces gibberellin and fusaric acid. The fusaric acid causes stunting and gibberellin causes elongation. Pathogen fusarium will produce micro and macroconidia along with the chlamydospores. Coming to taxonomy, this will belong to the phylum Ascomycota. These are the pictures representing microconidia and macroconidia of the fungus along with the perithecium. This perithecium is a flask-shaped fruiting body. The sexual stage of the fungus Gibberella fusicora will produce perithecium containing ascus and ascospore. These ascospores are usually two-celled ascospores. Coming to survival and spread of the fungus, the disease occurs most frequently when the infected seeds are used for sowing. Also can occur when the pathogen is present on the plant material or in the soil. That spreads through wind, water that carries the fungus from one plant to another. Buchanay disease can also be transmitted during farm operations such as harvesting the infected plants along with the fungal spore. It may mix with the healthy seed and it will cause severe infection. Also by soaking the seeds in a water that contains the fungal infection. Coming to management, one is clean. The seed should be clean sheath should be used to minimize the occurrence of the disease. Salt water treatment we can give. Those floating seeds we can remove, which are usually infected by the fungus. Seed treatment using fungicides such as Thyram, thiophanate methyl, benomyl is effective before planting. Benomyl or benlite application at the rate of 1 to 2% we can apply. Then seed treatment with organomercurial fungicide, agrofan, serozan. Usually these fungicides are not using nowadays, and also we can give application of copper sulfate solution, or you can also go for 2% formalin solution is recommended. So these are the different IDM practices for the control of Buchanai disease in rice. Then coming to important disease, bacterial disease in rice, which is bacterial blight of this bacterial blight of paddy, which is caused by the pathogen Xanthomonas variza, Pathover variza. It's said to be having the first seen by the farmers in the Fukuoka area of Japan in 1884. Yield loss due to bacterial blight can be up to 70% when there is a susceptible variety are grown. The first epidemic in India was reported from Shabad district of Bihar in 1963 and 64. Coming to the symptom, seedling wilt or kruzak is the main symptom. Seedling wilt or kruzak is it will occur during nursery stage, the sudden wilting and death of the young seedling. Water soaked to yellowish stripes on the leaf blade are starting at the leaf tips, then later increase in the length width with a wavy margin. Appearance of the bacterial ooze that look like a milky or opaque dewdrop on the young lesion during early morning hours. Lesion turn yellow to white the disease in as the disease advances. If the cut end of the leaf is kept in water, it becomes turbid because of the release of bacterial ooze or bacterial cell. Coming to the pathogen, Xanthomonas is a gram-negative pathogen with a single polar flagella. Bacterium is rod-shaped and it is aerobic in nature. And this belongs to the phylum Proteobacteria. It is a gram-negative. On the petroplate, it will produce the slimy yellowish colonies. And coming to disease cycle, mainly the pathogen bacteria will penetrate through adenoids present on the margin of the leaf during morning hours along with the moisture. Coming to favorable condition, clipping of the tip of the seedling at the time of transplanting, heavy rain, heavy dew, flooding, deep irrigation water, severe wind and temperature of 20 to 25 to 30 degree, application of excessive nitrogen, especially late tap dressing, which will all act as a favorable condition. Coming to management of bacterial blight of rice, we can use disease-free seeds for sowing, grow nurseries preferably in isolated upland condition, avoid clipping of seedling during transplanting, balanced fertilizer, avoid excessive nitrogen application, skip nitrogen application at boot leaf stage, drain the field except the flowering stage of the crop. Destruction of the wild collateral host, they will act as a collateral host for survival of the pathogen during off season. Avoid the flow of water from one field to another field, from infected to healthy fields. Coming to other management practice, grow tolerant varieties like IR20, TKM6, and we can also apply streptomycin sulfate or tetracycline in combination with copper oxychloride at the rate of 1.25 kg per hectare. So this will act as a antibiotic against the bacterial pathogen. So all these are IDM practices recommended to control bacterial blight of rice. Then coming to another important viral disease in rice, like rice tungro virus. This rice tungro virus caused by the viral pathogen. The causal organism, rice tungro bacilliform virus, the RTBV, it is containing double-stranded DNA as a genome, and rice tungro spherical virus, it is containing single-stranded RNA as a genome. Disease cycle, mainly primary infection, it occurs through some collateral host like Elisina indica, Echinoclova calanum, which will act as a collateral host for the viral infection. Secondary infection or secondary spreading mainly through leafhopper, nepotitic species, nepotitic Nigrocryptus, nepotitis virusins or nepotitis parvus. It will transmit the virus from one plant to another plant in a non-persistent manner. Coming to detection technique of rice tungro virus, tungro infected plants can be chemically identified by iodine test. Collect the leaf sample at 6 am. The top 10 centimeter portion of the leaf is immersed in a solution containing 2 gram of iodine and 6 gram of potassium iodide in 100 ml of water for 15 minutes, or 10 ml of tincture of iodine plus 140 ml of water for 1 hour. Then washed in the water for detection. Tungro infected leaves develop a dark bluish streaks. Then we can confirm the infection of rice tungro disease. Symptoms: plant affected by Tungro exhibit stunting and reduced tillering. Their leaves become yellow, orange yellow, may also have rust colored spots. Discoloration begin from leaf tip and extended down to the blade or lower leaf portion. Delayed flowering, panicles small and not completely exerted. Most panicles sterile or partially filled grains. So all these are the different symptoms, mainly that orange color leaves with a rusty color spots, which is the major identifying character of rice tungro virus. Coming to management, we can set up the light traps to monitor the attack of vector and destruction of the weed host, mainly present on the bands. And we can also grow tolerant varieties like CO45, CO48, Sureka, Vikramaria, or Barani, or IR36. In epidemic area, follow rotation with the pulse or oil seeds. And the greenleaf hopper vectors can be controlled by effective application of like monocrotophos, which is not available nowadays. Or you can also go for paspomidan or carbofuran granules or paspomidan or fenthian application. In nursery, when the virus infection is low, apply carbofuran granules at the rate of 1 kg active ingredient to control the vector population. During the pre-tillering to mid-tillering, when one affected hill is observed, apply carbofuran granules at the rate of 1 kg active ingredients. So these are some different IDM practices recommended to control the different diseases in rice tungro virus disease in rice. So this about important fungal, bacterial and viral disease in rice and their integrated disease management. Thank you.